Hello everybody, I'm Rob Reynolds. I'm the founder of Elbe Valley Medical. And as Alex mentioned, we're a micro-robotic cancer therapy. So every day we've got 22,000 people globally dying of cancer. Statistics show us that 1 in 4 of us will die of cancer at the moment. And we want to do something about that. We think it's fixable. So our purpose is to protect the people that we care about from cancer. It's that simple. So the niche for our therapy, we're looking at the largest and most aggressive tumors that are currently untreatable. Basically, any tumor with a necrotic core is ideal for our therapy, as long as it's a solid tumor, of course. And critically, we're coming in at a very, very low cost. So we would be providing this therapy in the United States for €6,000 per patient. It's a one-time treatment. And the way that the therapy works, I thought I had, yes, you lie on an MRI scanner, you get an injection intravenously into the arm, and you're placed in the MRI scanner. So during that time, we use this platform on the side to deliver RF to heat the necrotic tissue inside the body. And the way it works is rather simple. If this plays, the devices, they travel around in the blood, as you see here, with the red blood cells, they go everywhere. But inside the tumor with the necrotic core, the temperature rises to 41 degrees, and that's enough to trigger these microbots. So they open their arms, the cross-sectional size increases, and they can no longer fit through the blood capillaries. So what happens is they get stuck in the blood capillaries, and they embolize this tumor pretty quickly. So we're thinking about 30-minute treatment time, and the tumor is dead at the end. It's not dying, it's dead. The beauty of this treatment is, in conjunction with the MRI, we can both see the tumor, we can see the temperature of the tumor, but we can also see the accumulation of the microbots. So we can actually see the tumor die in real time in the MRI.
Wow.
We already have a preclinical trial done with mouse models in the University of Sheffield using 2 different tumor types. We've been able to prove safety even in an overdose condition. So we simulated a condition where 20 nurses would have injected a patient back to back, and the patient was put beside a large radio transmitter where everything was activated. There was no adverse effects seen even under that condition. And we were able to also demonstrate efficacy in both mouse types as well. So the devices themselves, they're quite small. You can see on the left there the size of a human hair and the size of our microbot by comparison. And to give you an idea of how much is injected into a human, that's a 1 milliliter of material on that teaspoon. And we'll be going for 1/16 of that. That's the dry volume of microbots that a patient would be injected with. So very, very small amount. And this is kind of what they look like when they're activated and unactivated. After the therapy, they cool down, the arms will close and the blood supply returns. So this is the most exciting slide. This is my favorite slide. This was taken from one of the mice in the preclinical trial. And what you're going to see in this is you're going to see these dummy microbots actually accumulate inside the tumor tissue. If this plays, you can see it here. These are the luminescent spheres that we used, and this here is actually the tumor tissue itself. Let's see. So yes, we were able to demonstrate hypoxia in this tumor tissue. This was within 24 hours. We didn't see any sign of necrosis with the first tumor type, but that's because we didn't wait long enough. We found out later from an oncologist we should have waited a bit longer, but we did see hypoxia. And so we're planning to do a further animal trial with the commercial product that we're looking to bring out. This will be the product that we want to bring to market. Our plan then is to immediately enter the veterinary market, because one of the big pieces of feedback we've gotten from potential investors is need to see revenue quickly, please. So we're doing this trial in France with dogs with naturally occurring cancers. And that will open the door, of course, to a phase 1 trial after that. So the veterinary market is about $1 billion between Europe and the US. And we already have a distributor appointed for the veterinary product. So we have some big names there: Fraunhofer Institute, Pyrexer Medical, etc. And we have a great team. There's myself, Douglas, our CEO, Dr. Shalja Kumar is the head of R&D.
Heather,
And
Heather, are we out of time?
Yeah, 5 minutes. Thank you. That was great.
Okay. So
okay, great.
So Rob, tell me about how you, just spend 30 seconds finishing. We'll let it go. Just
okay. So we'll be expecting to be cash generating by 2022. We have a gross margin of over 70%. And then we go into a little bit about the revenue because, you know, how we actually ended up with the revenue figures that we did. So to get to a revenue of 28.8 million.
It's okay. You can skip this part. I mean, it's all made up at this point anyway. Okay.
Alex.
Well, I mean, they don't have a product.
I know.
Okay, so Rob, you want to start out the questioning? Because I know you're chomping at the bit for this one.
Oh, I love this one. Is this shape memory alloys?
And no, we don't use.
Don't ask me to reveal anything critical, Rob. Come on.
What? Well, I wanted the question goes to: are these FDA rated materials? I've only saw CAD. I'd love to see an image of an actual one. And the follow-on question is going to be: do they get stuck, or how sure are they that you can actually get rid of them after the treatment?
And they're not going to get stuck in like other hotspots, like I don't know.
Yes.
Yeah.
Brain,
well,
mini strokes, this type of thing.
Yeah.
So,
yeah.
To answer the first question about the materials are mainly tungsten and PMMA, the polymer. The device then operates a little bit like a bimetal strip, so it isn't shape memory alloy. It's even simpler than that.
Okay.
The devices are basically stupid. There's no clever technology on board. We've kept it as simple as possible so that it's scalable and that it's low cost and it's easy to manufacture. The manufacturing process is from the 1990s, so it's been around a good while. So to answer the question about them getting stuck inside tissue, we simulated in the preclinical trial a similar situation to where a patient might be injected accidentally by 20 nurses back to back to get the dose up that high. And the devices we used were already activated. So we simulated where a patient was injected by 20 nurses and then placed beside a really strong radio transmitter. And we saw no adverse effects even under those conditions. So how did the devices come out? Well, we have designed them that they can stay in, but they can also be removed by filtering the blood with a magnetic filter. You pass the blood through a magnetic sieve and the devices will stick in the magnetic sieve.
Yeah, but you were talking about 41C. I mean, aren't there other parts of the body that will be at 41C, anisotropically?
So when you do, we're working with Pyrexer Medical. They have a treatment for heating tumors to make them more susceptible to radiation treatment and to chemotherapy. But the thing that's required is a necrotic core inside the tumor. And so it's a little bit like meat in the microwave. It heats up from the inside out. And so when our devices come near that tissue, they also embolize. And so the tumor dies from the inside out, and we can track that inside the MRI. Okay. Actually, both the accumulation of the devices inside the tumor and the increase in temperature of the tumor indicating that the tumor is dying.
Other than magnetic filtering, is there a biological method that these come out?
To make it really simple, it's a little bit like dialysis, except much cheaper. You tap into an artery, you pass the blood basically through a filter and back in through a vein, and the devices will remain inside that magnetic sieve.
Okay, questions from anybody other than Rob or I?
I have a question. Nowadays, especially for complex tumor, you do have a combination of both radiotherapy, surgery, and chemotherapy. Is the goal by using your technology to remove the three of them? You'll do a combination of everything.
It's to remove everything else because chemotherapy has nasty side effects and so does radiation therapy. So all we're doing is we expose the patient to RF, and we also allow the clinician to take more of a risk. At the moment, you need to err on the side of removing healthy tissue in order to remove all the cancer cells. But we're hoping we can move that story that a clinician can take more risk because a reoccurring tumor is equally treatable, where it isn't with traditional EBRT.
That's great.
I'm a little surprised with that answer, just because I would think that the tumor burden that there would be advantage of doing this early and then doing chemo for, especially if you've got microscopic seeding of the disease elsewhere, but not my specialty, just things I've heard about.
So that's correct. So we're on a personal level, I'm really interested in the late stage tumors, but the early stage tumors are equally valid. The reason why I'm interested in the late stage tumors is because there's nothing to treat in the late stage at the moment. And the burdens that we have to prove the therapy in that space are that much lower, if that makes sense. But you're correct. One of the ideal ways would be you take our treatment and then maybe a small round of chemo to clean up any cells that might be spreading through the body. But if that's not possible, our solution is wait till the tumors grow, come in on a Tuesday afternoon, get your therapy and go back to work.
I can imagine with some recurrent growth tumors that are, technically benign, but you can't really stop them. This could be something.
Last question.
Is this something that can be done on multiple tumor sites, or do you have to kind of do one at a time?
There's two configurations. One is where you can destroy all tumors with the necrotic core, but you can also just use them in dual heating mode, where you can use two beams that overlap, and you can use it for microsurgery. So you can use it to kill any tissue you want, not just cancer tissue in that mode.
Thank you. That was great.
Thank you very much.
Really fantastic job, Rob. Very, very exciting. And this is what we get into this for, right? I think going back to Rob and I, when his dad first was diagnosed with cancer, when we were in our 20s, this is exactly what we were looking for, something like this.
Did we miss something? Maybe I missed it. How much are we looking for to raise here?
Yeah, there wasn't a discussion of that. And also, what would you be investing in, since you have a bunch of other companies?
Well, everything. We're a company that builds companies. That's the way we